Today we're going to learn about the work Gregor Mendel did and how we apply his theory of particulate inheritance to our modern view of genetics. We'll also learn how to apply probability rules to solve genetics problems. Let's start by thinking about some of the ideas that were prevalent at the time of Mendel in the mid-1800s. One important theory was blending inheritance. According to this idea, offspring were a mixture of their parents, and their physical characteristics were essentially a blending of the two parents. Even though we now know that this isn't true, it's not an unreasonable hypothesis, and many experiments and our own intuition can be used to support it. For example, the height of children is often intermediate between the height of their parents. In some flowers, the color is intermediate between the two parents. You can see a problem with this, however, is that after a few generations, all of the variation in a population will be lost, as every offspring will become more and more average. As you'll learn in another course, variation is the basis of evolution, and the raw material on which evolution acts. If blending inheritance were to happen, all of this variation would disappear, and evolution would cease. Another idea that was common in the 1800s was that evolution happened through the inheritance of acquired characteristics. This was a theory proposed by a Frenchman, Jean-Baptiste Lamarck. His idea was that characteristics gained by an individual during its lifetime could be passed on to its offspring. So, in his famous example of giraffes, a giraffe that ate well and stretched its neck repeatedly to get at the higher branches of a tree could pass on that lengthened neck to their offspring. Again, this sounds like a crazy idea, but there was evidence that was used to support it. Birds like ostriches don't fly, birds like ostriches that don't fly lose their ability to fly and gain weight. Spadefoot toads lay eggs, and if those eggs grow up in environments with fairy shrimp, the tadpoles will become carnivorous and cannibalistic. But if they grow up in a pond without fairy shrimp, they'll be herbivorous and grow more slowly. Some offspring of the carnivorous spadefoot toads are born to be carnivorous even without the environmental stimulus. Although we know that inheritance doesn't work as Lamarck proposed, this case looks like Lamarckian inheritance of acquired characteristics because the offspring are directly inheriting the carnivorous traits of their parents. So these were some of the ideas that were floating around in the 1800s when Gregor Mendel did a series of experiments that showed that inheritance was particulate, or that there are discrete heritable units. Let's look at how he came to this conclusion. Mendel studied garden peas, which were very easy to grow. He developed true breeding varieties. That is, he developed varieties that when self-fertilized only ever produced plants with the same characteristics. For example, true breeding purple flowers would only ever produce other purple-flowered plants when self-fertilized. Although there were many characteristics of pea plants that he could have chosen, he chose to study characteristics for which there were only two traits. For example, flowers were either purple or white, seeds were either yellow or green, and they could either be round or wrinkled. The seed pods could be green or yellow, and they could have either inflated or constricted shape. These traits were easy to classify, and there were no intermediate traits that would have made classification difficult. Mendel did his experiments by cutting off the stamens, or the male parts, from one plant so that the plants could not self-fertilize. He used pollen from another plant to fertilize the female parts of the original flower. After fertilization, he grew the seeds until they were mature and planted them to observe the offspring, or the first filial generation. In one of Mendel's experiments, he crossed true-breeding purple flowers with true-breeding white-flowered plants, and the results were an F1 generation of all plants having purple flowers. Mendel then took the purple flower F1 generation and selfed them to produce an F2 generation, and he noticed that there were about 3 times as many purple-flowered plants as white-flowered plants. Interestingly, when he reversed the sex of the parents, he obtained exactly the same results. One of the first things to notice is that there's no blending inheritance going on here. The purple and white flowers produced purple flowers, not something in between. The second important thing to note is that there is something that Mendel called a heritable factor that had been carried through the F1 generation and expressed again in the F2 generation. This heritable factor was not lost in the F1 generation. It carried through into the F2. The last important thing to notice is that Mendel did this experiment quantitatively. That is, he was very careful to count all of the plants, all of the offspring in each generation. Through these careful experiments, and many others like it, Mendel noticed that there was always a 3-to-1 ratio in the F2 generation, with 3 times as many individuals of one trait than the other. We now know that the particulate inheritance that Mendel noticed is due to the presence of different alleles on particular genes. Genes are stretches of DNA that code for a protein. The exact genetic code required to make the protein is encoded in this stretch of DNA. However, there may exist more than one sequence to make the protein, and some of those sequences may produce a somewhat altered or even non-functional protein. We call the 2 versions of the gene alleles. In a diploid organism, the presence of 2 alleles will determine what the trait looks like. In an individual that's true breeding, we now say that it is homozygous. This means that the 2 copies of the genes in the diploid individual, or the 2 alleles, are the same. If the 2 alleles are different from one another, then we say the individual is a heterozygote. This individual shown here is a heterozygote. It has inherited one allele from its father, shown by the gene on the blue chromosome, and one allele from its mother, shown by the gene on the red chromosome. Because there are 2 different alleles in this individual, it's a heterozygote. The allele it's inherited from its father codes for an enzyme that's able to synthesize the purple pigment used in the flower. The allele inherited from the mother is slightly different and as a result does not successfully code for that enzyme. As a result, no pigment is produced by that allele. In this case, the presence of one functional allele that can produce the enzyme allows even a heterozygote to produce enough purple pigment to make the flower purple. As a result, we say that the purple color is the dominant trait. Heterozygotes are purple, and the dominant allele hides the recessive allele. The only way that we can see the presence of the recessive allele is if we have a homozygote with 2 copies of the recessive allele. This is the case for white flowers. They have 2 copies of the recessive allele, and neither one of them produce functional enzymes, and therefore no purple pigment is produced. We give alleles names, and one of the conventions is to give the dominant allele a capital letter and the recessive allele a lowercase letter. Thus, we could give the allele on the blue chromosome a capital P and the allele on the red chromosome a lowercase p. Okay, before we go on, let's remind ourselves of what happens in the production of gametes, or sex cells, by meiosis. Remember that if we have a pair of homologous chromosomes, they duplicate in S phase, and then the homologous chromosomes, the homologs, separate in meiosis 1, and the sister chromatids separate in meiosis 2. The result of meiosis is 4 gametes, and each of these gametes has only one of the original 2 chromosomes. Therefore, if there's a gene along that chromosome, 2 alleles will be divided up with 1 allele in each of the haploid daughter cells. Okay, so here we have a purple flower being mated with a white flower. The purple flower is a homozygote and therefore has 2 purple alleles. When cells go through meiosis, these 2 purple alleles will separate into the different gametes. However, since they're the same, we simply note that this plant can only produce purple alleles, shown by the uppercase P. The white flower is also a homozygote, but has 2 white alleles, shown by the lowercase p, and therefore can only produce gametes with the white allele. When fertilization happens, A gamete with a big P fertilizes a gamete with a little p to create a heterozygote, big P, little p. Notice that all of the individuals in the F1 generation are heterozygotes. When these are self-pollinated, the heterozygotes produce gametes with either the purple big P allele or the white small p allele, and they produce these gametes in equal numbers. If a gamete containing the big P allele meets another gamete containing a big P allele, they fertilize one another and the offspring will have two big P alleles, and therefore be a big P homozygote. If a big P allele meets a little p allele, the resulting offspring are big P, little p heterozygotes. Finally, if a little p allele meets another little p allele, the offspring will be little p and will be white. This way of setting this up in a chart like this is called a Punnett square. The gametes produced by the parents always go on the outside of the square. The inside of the square indicates the offspring. You can see that there are two ways to get heterozygote offspring. Either the male parent can contribute to little p and the female parent can contribute to big P, or the other way around. You can also see here that since the big P and little p gametes are produced in equal numbers by meiosis, we'd expect to see 3 times as many purple flowers as white flowers, and this is exactly what Mendel found. So you can see that the alleles of a gene underlie the appearance of a trait. In this case, we have purple and white flowers. We call this the phenotype, or the appearance of a trait. The combination of alleles that gives a plant its appearance is called its genotype. Now, because the purple allele is dominant, any homozygote with two purple alleles will be purple. Additionally, a heterozygote with a purple and a white allele will be purple because the purple allele is dominant and will essentially hide the white allele. The only genotype that can produce a white-flowered plant is a homozygote for the white allele. So, their phenotype describes the appearance of the plant, and the genotype describes which alleles it has. You can describe the genotype either by directly describing the alleles or by indicating the alleles by way of saying it's homozygous or it's heterozygous. Notice that the ratio of phenotypes is 3:1. That is, there are 3 purple flowered plants for every plant with white flowers. The genotype ratio, however, is 1:2:1, with there being 1 homozygous dominant plant for every 2 heterozygotes for every 1 homozygous recessive plant. Be careful to always give the correct ratio if you're asked to give either the genotype or phenotype ratio. Okay, let's do another Punnett square just for practice. Pea seeds can either be round or wrinkled. We indicate the round allele with an uppercase R because it's dominant, and the wrinkled allele with a lowercase r because it's recessive. To start with the Punnett square, we need to assign the gametes to the edges of the square. The big R homozygotes can only produce big R gametes. The big R little r heterozygotes produces half big R gametes and half little r gametes. Let's go ahead and have the gametes fertilize one another to produce offspring. Notice that the offspring all have round seeds, but there are 2 genotypes possible that both produce that round-seeded phenotype. Finally, notice that you can simplify this because there's only one type of gamete produced by the homozygote. Therefore, it's easier to see the 50-50 genotype ratio like this. If we have a plant for which we don't know its genotype, we can use a technique called a test cross in order to determine what its genotype is. First of all, if the plant has the recessive phenotype, then we immediately know its genotype. It must be a homozygous recessive, or in this case, little p. However, if it has the dominant phenotype, purple in this case, we don't know if it's a homozygous dominant big P, or a heterozygote. We can use a test cross to figure this out. In a test cross, we cross an individual with an unknown genotype against a tester and look at the offspring. The tester is a homozygous recessive. If the unknown plant was a homozygote, all of the offspring of that cross would be heterozygotes, and we have the dominant phenotype. If, however, the unknown individual was a heterozygote, half of the offspring will be expected to have the recessive phenotype, and half will have the dominant phenotype. Therefore, you can do this experiment, and if you get offspring with the recessive trait, you know that the unknown parent is a heterozygote. Let's expand what we've been doing with one gene and now track two genes. In this case, we're tracking a gene that makes the seed yellow or green, with yellow being dominant, and we're also tracking a gene that causes the seeds to be either round or wrinkled, with round being dominant. Remember that pea plants are diploid, and so for every gene, they have two copies of the gene, and these may be the same or they may be different alleles. Let's start out with a yellow, round homozygote and cross it with a plant with wrinkled green seeds. When each of these plants produce gametes, remember that the gametes will have only one copy of each gene. When the gametes fertilize each other, we get an F1 generation. That's a dihybrid. That is, it's a heterozygote at both genetic loci. Let's see what happens when the F1s mate. There are actually two possible hypotheses as to how this could happen. In the first hypothesis, imagine that the two genes are inherited together as a single unit. If this is the case, then the gametes produced by the F1 will either be big Y big R or little y little r. If this happens, then we would expect to get a 3-to-1 phenotypic ratio, with 3 round yellow peas to 1 green wrinkled pea. The other hypothesis is of independent assortment. In other words, is the Y gene being inherited completely separately and independently from the R gene? If the two genes are separate, then each parent would be able to produce gametes with any of these combinations, and each would be produced in equal proportion. If there is independent assortment, then we would expect to get these genotypes and phenotypes. We would expect to have 9 out of the 16 offspring be round and yellow, 3 of the 16 offspring would be round and green, 3 would be yellow and wrinkled, and 1 would be green and wrinkled. This phenotypic ratio is 9 to 3 to 3 to 1. When Mendel did his experiments, he did in fact find a 9 to 3 to 3 to 1 ratio, suggesting that there is in fact independent assortment. Let's look a little bit in more detail about how independent assortment happens. Let's start by assuming that the two genes are located on different chromosomes. During meiosis I, the chromosomes will line up along the metaphase plate, and with two chromosomes, there are two different ways in which they can align. In the cell on the left, both maternal chromosomes are on the left and the paternal chromosomes are on the right. After separation of the homologous chromosomes, and later the sister chromatids in meiosis II, the gametes formed will have big Y big R and little y little r. If, just by chance, the chromosomes align differently during metaphase I, as on the right, the maternal chromosomes are mixed up with the paternal chromosomes in the final gametes. These final gametes would have big Y little r or little y big R. That is, all 4 types of gametes are produced, and furthermore, they're produced in equal proportions. This is the basis for independent assortment. In other words, the inheritance of one gene does not affect the inheritance of the other. They assort independently of one another. Okay, let's look at another dihybrid cross because many students have problems setting them up. When we set it up, we have to think about what gametes are possible. The way I do this is to start with the left allele of the first gene and choose that. Then I choose one of the alleles on the other gene and write that down. For the second one, I stay on the first allele of the first gene and move to the second allele of the second gene and write that down. Then I move to the second allele of the first gene and repeat. You can also change the order around and do it like the FOIL method in algebra. It's up to you. The important thing is that each allele for each gene has an equal chance of ending up in a gamete with any allele from the second gene. One common mistake I see students make is to not practice this when they get to an exam. They try setting it up something like this. This is a mistake because you're saying that a particular gamete only has one copy of the gene rather than one copy of each gene. I strongly encourage you to try setting up a 2-gene Punnett square. It isn't as easy as seeing it presented to you, and you're likely to make a mistake the first time around. Okay, this is the result of the cross we're looking at. Notice that I've just filled in all the interior squares with the gametes from the gametes on the edges. You can then go through and locate each square that has one of the 4 phenotypes. You should find that there are 9 squares with both traits dominant. There should be 3 squares with one dominant trait and one recessive trait. Finally, there should be 1 square with both recessive traits. This cross isn't a dihybrid cross. The first individual is a homozygote for the R locus. Therefore, we can simplify the Punnett square, and we don't need to show the whole thing. If you want to, however, it would be fine. One important thing to notice is that not all 2-gene crosses are dihybrid crosses. You need to pay special attention to the genotypes of the parents. Okay, we can actually do a lot of genetics using simple probabilities. Before we get into genetics, however, we need to cover 2 simple probability rules. The first rule is called the AND rule, or multiplication rule, and we use this rule when 2 independent events must both be true. If that's the case, then we multiply their probabilities. For example, what's the probability of getting 2 heads when flipping 2 coins? Well, this is like saying the first flip must result in heads and the second flip must result in heads. Both of those independent events must be true, so we multiply their probability. The probability of getting 1 head is 1/2, so we multiply 1/2 times 1/2 to get 1/4. The second rule is the addition rule, or the OR rule. We use this when only one of 2 or more mutually exclusive events will occur. When this happens, we add their probabilities. For example, what's the probability of getting a heads or tails when flipping 1 coin? Well, you must either get a heads or a tails, so these are mutually exclusive events. The probability of getting a head is 1/2, and the probability of getting a tail is also 1/2. So you must either get heads or tails. So we add these probabilities to get 1/2 plus 1/2 equals 1. This makes sense, since the probability is 1 that you get either heads or tails. Here's a slightly harder one. What's the probability of getting exactly 1 head when flipping 2 coins? To solve this, we have to realize that there are 2 ways to get 1 head. In the first way, the first flip is a heads and the second is a tails. The probability of this combination is 1/2 times 1/2. We use the AND rule and multiply these together. In the second way, the first is tails and the second is heads, and so the probability of that is also 1/2 times 1/2. In order to get exactly 1 head, either the first scenario is true or the second scenario is true, so we add those probabilities to get a total of 1/2. If both of them or neither were true, we would end up with more or fewer heads than 1. Let's apply these rules to heredity. For a really simple example, let's consider the probability of having big A offspring from a monohybrid cross as shown here. In order for the offspring to have 2 big A alleles, they need to inherit a big A from the mother and a big A from the father. We can write this mathematically using the AND rule, and so we multiply the probability that the male parent contributes a big A and the female parent contributes a big A. The answer to this is 1/2 times 1/2 equals 1/4, which we already knew from the Punnett square. Okay, Here's another simple one that incorporates both rules before we move on to something more difficult. What's the probability of having a heterozygote offspring from that same cross? In order for the offspring to be heterozygous, they must inherit a big A from one parent and a small a from the other parent. There are two ways to do this. They could inherit the big A from the mother and the little a from the father, or they could inherit the little a from the mother and the big A from the father. The probability of the big A from the mother and the little a from the father is 1/4, and the probability of the little a from the mother and the big A from the father is also 1/4. We add these together using the OR rule to get a probability of 1/2. Okay, here's a slightly harder one. What's the probability of having big A, big B offspring in a dihybrid cross? In this case, we can use the fact that the two genes assort independently, and we can therefore multiply the probabilities of having particular offspring at each gene. We can therefore view this as what is the probability of having a heterozygous offspring at the A locus and a homozygous big B at the B locus. The probability of getting a heterozygous big A is 1/2, and the probability of big B is 1/4. We multiply these to get 1/8, which you can check on a dihybrid Punnett square. When you solve problems like this, you do need to be careful to look carefully at the genotype of the parents. If we change the previous question ever so slightly by having a big B homozygote father rather than a heterozygote, then the question is different. Instead of the probability being 1/4 of getting a homozygous offspring, the probability is now 1/2 because the parent is now a homozygote. We can use probability to solve quite difficult problems. For example, what is the probability of having offspring that is homozygous at exactly one gene locus for this cross here? Notice that this cross is not a trihybrid cross because the father is a homozygote at the C locus. We start by writing down all of the possible genotypes of the offspring that we are interested in. The first one is homozygous at the A locus for the dominant trait. The second one is homozygous for the recessive trait. The third and fourth are homozygous at the B locus, and the fifth and sixth are homozygous at the C locus. If we write down each probability for each locus, then we can add them up at the end using the OR rule. Notice that the probability in the last case is 0, because there's no way to have offspring that is homozygous for the recessive trait at the C locus. If we add all these up, we get a total of 6/16, or 3/8 probability. I want to end with a warning about some particularly tricky cases. Let's look at the probability of having offspring that's homozygous at least one of two gene loci for this dihybrid cross. You might be tempted to say, oh, this is easy. All I need to do is figure out the probability of being homozygous at each locus and ignore the other locus. So you might say the probability of being big A and anything at the big B at the B locus is 1/4. Similarly, the probability of being a small a homozygote is also 1/4. If you did this for all of them and then added them up, you would see that you would have a probability of 1 of being homozygous at least one locus. However, unfortunately, this would be wrong. The reason this is wrong is that you would end up double counting some genotypes. For example, the big A, big B homozygote has been included in both the first and the third rows. We could list all of the genotypes that we double count and subtract them from the total. However, there's an easier way. In this particular case, we could rephrase the question, and instead of looking at for the probability of having offspring that's homozygous at least one locus, we could figure out the probability of having heterozygous offspring at all loci, and subtract this from 1. If we do this, we can calculate the probability of being heterozygous at all loci to be 1/4, and subtracting this from 1 leaves us 3/4. You can check this result by looking at how many genotypes we double counted in the first incorrect method. It may seem that using these probability rules can be quite tricky. However, you can solve much more complicated problems than you could with a Punnett square. A Punnett square that's larger than 2 loci becomes very unwieldy, as you would need 64 squares to accommodate a trihybrid cross and 256 squares for 4 loci.